Welcome to the deep dive. Today, we're really getting into the nitty-gritty of modern pathology. It's all about understanding disease right down to the molecular level within our cells.
Exactly. We're looking at how tiny disruptions in genes, proteins, metabolic pathways can cascade into actual disease states, how cellular behavior changes.
And our aim today isn't just a quick overview. We want to dig deep to really unpack the core principles and some of the latest breakthroughs, give you that actionable knowledge.
Right. Think of it less like a lecture, more like a deep exploration. We're building a framework here, a way to really grasp disease mechanisms for future discussions. It's about getting that solid foundation fast.
Absolutely. And you can't talk about this molecular revolution without mentioning the Human Genome Project. Sequencing our DNA back around the turn of the century, that was just the starting gun, wasn't it?
Oh, completely. It opened the floodgates, sequencing costs plummeted, and our ability to analyze all that data just exploded. Genomics, proteomics, metabolomics, these fields came into their own.
Giving us this incredible view of what our cells are actually doing.
Precisely. And what's become so clear is that the simple ATCG sequence is only part of the story. It's incredibly complex, the regulation, the interactions. We're still just scratching the surface.
But the potential there for understanding disease, for developing new treatments, it's immense. It feels like we've gone from a basic blueprint to understanding the complex operating system.
That's a great analogy, and it brings up a really interesting point. Humans, we have about 20,000 genes that code for proteins. Seems like a lot, but it's only about 1.5% of our genome.
Only 1.5%? That's tiny.
It is, and here's the kicker. Simpler organisms, like a C. elegans worm, much smaller genome, far fewer cells, they have a similar number of protein-coding genes. Many even look like ours.
So what makes us so different then, if it's not just the number of protein parts?
Well, that's the million-dollar question, isn't it? And the answer seems to lie overwhelmingly in the other 98.5% of our genome, the stuff we used to call, well, junk DNA.
Which we now know isn't junk at all.
Not even close. We know over 85% of the genome gets transcribed into RNA. And maybe 80% is involved in regulating those protein-coding genes. It's not just the building blocks, it's the incredibly sophisticated instruction manual for using them.
Okay, so it's all about the regulation, the orchestration. Let's break down this non-coding instruction manual. What are the key parts?
We can think of it in about 5 major categories. First, you've got promoter and enhancer regions. These are like the control switches for genes.
Promoters near the gene start, enhancers maybe further away.
Exactly. Promoters are usually right upstream, essential for getting transcription started. Enhancers can be thousands, even hundreds of thousands of base pairs away. They act like volume knobs, boosting expression, and they can actually loop through space to connect with the promoter.
Wow, like a physical connection across a long distance. Okay, what's next?
Second, there are binding sites for proteins that organize chromatin. Chromatin is how our DNA is packaged with proteins in the nucleus.
Yeah.
How it's folded really matters for which genes can be read.
Right, accessibility is key. So these sites help manage the physical structure.
Got it. Third
are the non-coding regulatory RNAs. These are RNAs that don't make proteins themselves, but act as regulators. The big players here are microRNAs, or miRNAs, and long non-coding RNAs, LNC-RNAs. We'll definitely dive deeper into those.
Okay, RNAs that regulate other genes. Fourth category.
Fourth, we have mobile genetic elements, transposons, or jumping genes. These sequences can actually move around the genome.
Jumping genes. Sounds a bit chaotic.
It can be. They make up a surprisingly large chunk of our DNA. We're still figuring out all their roles in regulation and chromatin structure, but they're definitely not just sitting there.
Interesting. And the last category.
The fifth includes specialized structural regions. Think telomeres, the protective caps on chromosome ends that stop genes from degrading during replication. And centromeres, the bit where chromosome copies link up and where the spindle fibers attach during cell division. They contain lots of repetitive DNA, satellite DNA, and help organize dense, inactive chromatin. It's really telling that so many genetic variations linked to diseases are found in these non-coding regions. It just hammers home how crucial gene regulation is for health.
Absolutely. And think about this. Any two people are more than 99.5% identical in their DNA sequence. We share about 99% with chimps.
That tiny difference, less than 0.5%.
Which is still about 15 million base pairs, accounts for all human diversity. Susceptibility to disease, response to drugs, everything. It shows how impactful even small variations in the blueprint can be.
Puts things in perspective. So within that small variation, what are the most common types we see?
2 main types dominate. SNPs, single nucleotide polymorphisms, and CNVs, copy number variations.
SNPs are just single letter changes, right?
Essentially, yes. A variation at one specific spot in the DNA sequence. Usually there are only two versions or alleles in the population. We know of millions of them.
And their frequency varies between populations.
It can, yes. Most SNPs are actually outside of protein coding regions. But if a SNP falls on a promoter or enhancer, it can change how regulatory proteins bind, affecting gene expression. Some seem to do nothing. They're neutral.
But even neutral ones can be useful.
Definitely. They can act as markers. If a neutral SNP is physically close on the chromosome to a gene variant that does cause disease, they tend to be inherited together. That's called linkage disequilibrium. So we can use the SNP as a flag to find the region where the actual culprit gene lies.
Like signposts on the genome map.
Okay.
And CNVs, copy number variations.
These are much larger changes. Segments of DNA anywhere from 1,000 to millions of base pairs long that are present in different numbers of copies between individuals. You might have extra copies or deletions.
So big chunks duplicated or missing, that must have a significant impact.
It really does. It accounts for a lot of genetic difference between people, maybe 5 to 24 million base pairs worth. And about half of these CNVs actually involve protein coding genes. So changing the dose of a gene can have major effects on traits and disease risk.
Okay, so SNPs and CNVs explain a lot of the sequence variation. But that still doesn't explain everything, like differences between identical twins.
Which brings us to epigenetics, right?
Exactly. Epigenetics, this is a huge area. It refers to changes in gene expression that are heritable passed on during cell division, but that don't involve changes to the actual DNA sequence.
So it's like a layer of control on top of the DNA sequence.
Precisely. It's how different cell types in your body, all with the same DNA blueprint, end up looking and functioning so differently. Liver cells, neurons, skin cells, they run different programs using the same genetic code, largely thanks to epigenetic factors. It's about fine-tuning gene activity.
Okay, so if it's not the sequence, what are these epigenetic factors doing? How do they work?
A major part involves how DNA is packaged. Our DNA wraps around proteins called histones, forming structures called nucleosomes like beads on a string. This whole complex is chromatin.
Right, the DNA protein complex. And histones aren't just passive spools. They can be chemically modified. Enzymes called chromatin writers add little chemical tags marks to the histones. Things like methylation, acetylation, phosphorylation. Over 70 different marks known.
Like little Post-it notes on the DNA packaging?
That's a good way to think of it.
Yeah.
And these marks influence how tightly the chromatin is packed. Open, accessible chromatin euchromatin tends to have marks associated with active genes, allowing the transcription machinery access. Tightly packed chromatin heterochromatin has marks associated with silenced genes.
And these marks can be changed.
Yes, that's key. There are chromatin erasers that remove the marks, and chromatin readers proteins that recognize specific marks and recruit other machinery to regulate gene expression. It's dynamic.
Can you give some examples? Like, what does methylation do?
Histone methylation, adding a methyl group to lysine or arginine, can activate or repress genes depending on which amino acid gets the mark and where it is. For example, methylation on lysine 4 of histone H3, H3K4, Me3 is usually at active genes. Methylation at lysine 9, H3K9Me3, is often found in silenced heterochromatin.
Okay, and acetylation.
Acetylation, adding an acetyl group to lysine, done by histone acetyltransferases, or HATs, generally loosens chromatin structure, making genes more accessible and boosting transcription. Histone deacetylases, HDACs, remove acetyl groups, condensing chromatin and silencing genes. Phosphorylation adds phosphate groups to serines, and that can also open or close chromatin, depending on the context.
It's not just the histones being marked, right? The DNA itself can be methylated.
Correct. DNA methylation, adding a methyl group directly to a cytosine base, especially in promoter or enhancer regions, usually acts as a silencing signal, shutting down gene transcription. This is controlled by DNA methyltransferases, writers, demethylating enzymes, erasers, and proteins that bind to methylated DNA to mediate the silencing.
And there are also factors organizing the larger structure.
Yes, chromatin organizing factors. We're still learning about them. But they seem to bind specific non-coding DNA regions and control the long-range looping of DNA, bringing distant enhancers close to promoters to activate genes.
Wow. It's an incredibly complex regulatory network.
It really is. How all these epigenetic layers interact to control genome organization and gene expressions specifically in each cell type is a huge area of research. We do know that messing up the epigenome is fundamental to cancer, and there's growing evidence linking it to many other diseases too.
But the hopeful part is These epigenetic marks aren't necessarily permanent like DNA mutations.
That's a critical point. Many are reversible. Histone acetylation, DNA methylation enzymes can add and remove these marks. This makes them really attractive targets for therapies. Drugs that inhibit HDACs or DNA methylation are already in clinical trials, especially for cancer, trying to kind of reset abnormal gene expression patterns.
Okay, shifting gears slightly, but still in the realm of regulation. Let's talk about those non-coding RNAs you mentioned, microRNAs and long non-coding RNAs.
Yes, another fascinating layer of control. These RNAs are transcribed from DNA but don't get translated into proteins. They function as RNA molecules themselves to regulate gene expression.
Let's start with microRNAs, miRNAs. What do they do?
miRNAs are tiny RNA molecules, usually only about 22 nucleotides long. They act as post-transcriptional regulators, meaning they don't stop a gene from being transcribed into messenger RNA, mRNA, but they interfere with that mRNA being translated into protein, or they target the mRNA for degradation.
So they silence genes after the initial RNA copy is made.
Exactly. It's a fundamental mechanism found in pretty much all eukaryotes. And because they affect protein levels, they're critical regulators of development, normal cell function, and also pathology, like cancer.
How many are there?
The human genome encodes somewhere around 6,000 different miRNAs. That's a huge number, maybe 30% of the number of protein-coding genes. And a single miRNA can often target hundreds of different mRNAs.
So one small RNA can orchestrate a whole program of gene expression.
Precisely. It's incredibly efficient.
How are they made and how do they find their targets?
It starts with the gene being transcribed into a primary miRNA, primRNA. This gets processed in the nucleus into a shorter hairpin structure. The precursor miRNA, pre-miRNA, that's exported to the cytoplasm, where an enzyme called dicer trims it into the mature, short, double-stranded miRNA. One strand of this gets loaded into a protein complex called RISC, the RNA-induced silencing complex.
And RISC is the actual silencing machine.
Yes. The miRNA acts as a guide. It base pairs with a complementary sequence, usually in the 3' untranslated region, 3' UTR, of target mRNA. If the match is perfect or near perfect, RISC often cleaves the mRNA, destroying it. If the match is less perfect, RISC usually just blocks translation, preventing the protein from being made. Either way, the target gene is silenced.
And what about SIRNAs? Small interfering RNAs.
SIRNAs are similar, also short, double-stranded RNAs processed by dicer and loaded into RISC. But they usually come from outside the cell, maybe from viruses, or often they're introduced experimentally by researchers. They typically have a perfect match to their target mRNA and lead to very efficient cleavage and degradation.
So they're like synthetic miRNAs we can use in the lab.
Exactly. They're powerful tools for knocking down specific genes to study their function. That's RNA interference or RNAi. And there's huge interest in using synthetic SIRNAs as drugs to silence disease-causing genes, like oncogenes.
Fascinating. Okay, now on to the long non-coding RNAs, LNCRNAs. You said there might be even more of these.
Current estimates suggest there could be tens of thousands, maybe 10 to 20 times more LNCRNA genes than protein-coding genes. It's a vast, largely unexplored territory. They're defined as being longer than 200 nucleotides and not coding for proteins.
And they regulate genes in different ways than miRNAs.
Yes, LNCRNAs are incredibly diverse in their mechanisms. Some bind directly to chromatin, physically blocking access for the transcription machinery. The classic example is XLEST RNA, which coats one X chromosome in females and shuts it down for dosage compensation.
A chromosome-wide silencer. Wow. What else?
Some act as decoys, binding transcription factors so they can't bind to DNA. Others act as guides, recruiting chromatin-modifying enzymes like HATs or methyltransferases to specific gene locations to turn them on or off. And some act as scaffolds, bringing multiple proteins together into functional complexes that influence chromatin structure or gene activity.
So they can be blockers, decoys, guides, or scaffolds. Very versatile.
Very. And interestingly, many enhancers, those distant regulatory elements, actually produce LNCRNAs. These eRNAs might themselves help activate the target genes.
It paints a picture of just layers upon layers of regulation. Okay, for our final topic in this section on the genome and its control, let's talk gene editing, specifically CRISPR.
Ah, yes. CRISPR-Cas9. Truly revolutionary. It gives us the ability to make precise changes to the DNA sequence in living cells with unprecedented ease and accuracy. And it came from a really unexpected place, bacterial immunity.
Bacteria have immune systems. How does that work?
They do, an adaptive one, in fact. They can remember viruses, bacteriophages that have infected them before. They incorporate little snippets of the virus DNA into their own genome, into these special regions called CRISPR arrays.
Okay.
These viral snippets are then transcribed into short guide RNAs, gRNAs. These gRNAs team up with a nuclease enzyme like Cas9. If the same virus infects the bacterium again, the gRNA recognizes the viral DNA sequence and guides Cas9 right to it. Cas9 then cuts the viral DNA, disabling it. It's a targeted defense system.
So how did scientists turn that into a gene editing tool?
They basically hijacked the system. We can now design custom gRNAs that match almost any DNA sequence we want to target in, say, a human cell. We introduce that gRNA along with the Cas9 enzyme into the cell.
And the gRNA guides Cas9 to that specific spot in our DNA.
Precisely. Cas9 then makes a clean, double-stranded break right at that target site.
And then what happens? The cell has to fix that break.
Exactly. The cell's own DNA repair machinery kicks in. There are two main ways it does this. One is called non-homologous end joining, NHEJ. It's quick but messy. It just sticks the broken ends back together, often introducing small insertions or deletions in the process. These errors can effectively knock out the gene if the break is within a coding sequence.
Okay, so NHDJ is good for disrupting genes. What's the other way?
The other pathway is homology-directed repair, or HDR. This is much more precise. If we provide the cell with a donor DNA template that has sequences matching the regions around the break, the cell can use that template to repair the break accurately. And crucially, we can design that donor template to include specific changes, correcting a mutation, or even inserting a whole new piece of DNA.
Incredible. So you can either knock out a gene or precisely edit it using the cell's own systems, and it's much more specific than older methods.
Massively more specific. Cas9, guided by a well-designed gRNA, is remarkably accurate, much better than older tools like zinc finger nucleases or talons. This reduces the risk of making unintended cuts elsewhere in the genome off-target effects, which was a big concern.
And the applications are huge, I imagine.
Absolutely enormous. Basic research uses it constantly to create cell and animal models of disease by introducing specific mutations. It's sped up the creation of transgenic animals immensely. Therapeutically, the potential to correct genetic mutations for inherited diseases is staggering. And of course, it raises significant ethical questions, especially around editing the germline or enhancing traits.
A powerful tool, definitely, with profound implications. Okay, we've covered the genome, epigenetics, non-coding RNA, gene editing. Let's shift now to the fundamental housekeeping tasks every cell needs to perform just to stay alive.
Right, these are the basics. Every cell needs to protect itself, get nutrients, metabolize them, communicate, sometimes move, constantly renew its parts, get rid of waste, and generate energy. The absolute essentials.
And these functions are largely compartmentalized within organelles, these membrane-bound structures inside the cell. Why is that important?
Compartmentalization is key. It allows the cell to concentrate specific enzymes and substrates together, making reactions more efficient. It also lets the cell isolate potentially harmful processes. Think about the digestive enzymes and lysosomes. You wouldn't want those loose in the cytoplasm. And it creates unique environments, like the low pH in lysosomes or specific ion concentrations in other organelles optimized for particular functions.
Okay, let's take a tour of the key organelles, starting with the cell's outer boundary, the plasma membrane.
Right. The plasma membrane, and indeed all the internal organelle membranes, are much more than simple barriers. They're fluid bilayers made mainly of phospholipids. These molecules have a water-loving head and water-fearing tails, thus they naturally form a barrier in water.
Letting some things pass but blocking others.
Exactly. Small, nonpolar molecules like O2 and CO2 slip through easily. Steroids too. But it's a barrier to large molecules, and especially charged ions. And crucially, the membrane isn't uniform. It's a mosaic of different lipids, and their distribution isn't random. It varies between the inner and outer layers in different regions. This lipid composition is vital for function, influencing how membrane proteins work and where they are.
Can you give some examples of important phospholipids?
Sure. Phosphatidylinositol is mostly on the inner side. It can get phosphorylated to create docking sites for signaling proteins, or it can be cleaved to generate second messengers like IP3 and DAC, which are crucial in signaling. Phosphatidylserine is also normally on the inner leaflet. But if a cell is undergoing apoptosis, programmed death, it flips to the outside.
Acting as an eat-me signal.
Precisely. It tells phagocytic cells to clear it away. It's also important on platelets for blood clotting. Then you have glycolipids and sphingomyelin, mostly on the outer surface. They contribute to the glycocalyx, the cell's sugar coat, and are involved in cell recognition, like immune cell recruitment.
And you mentioned lipid rafts, specialized zones within the membrane.
Yes. Even though the membrane is fluid, certain lipids, like single lipids and cholesterol, can cluster together, forming these more ordered microdomains, lipid rafts. Proteins might prefer to be in or out of these rafts, so rafts help organize proteins, affecting signaling, cell interactions, and how vesicles bud off or fuse.
And of course, the membrane is packed with proteins doing all sorts of jobs.
Absolutely. Transporting things across, receiving signals, attaching to other cells or the extracellular matrix. How they associate with the membrane reflects their job. Some span the whole membrane, transmembrane proteins, often for transport or signaling. Others are anchored by attached lipids or linked non-covalently to transmembrane proteins.
And they can form complexes.
Yes, often they work together in large complexes. And their distribution isn't random either.
Yeah.
They can be confined to specific domains like lipid rafts or areas defined by cell junctions like tight junctions. This helps create cell polarity, like the difference between the top apical and bottom sides basolateral of an epithelial cell. And don't forget the glycocalyx on the outside that sugarcoat provides protection and is involved in cell interactions.
So a very dynamic and organized boundary. But if it's a barrier, how do essential things like nutrients and ions get across?
That's where membrane transport proteins come in. Small nonpolar stuff diffuses, as we said. Water can diffuse, but specialized channels called aquaporins speed it up dramatically where needed, like in the kidneys. But for larger polar molecules like glucose or any ions, you need help.
Carriers and channels.
Exactly. Two main types. Channel proteins form pores, specific for certain ions or small molecules, allowing rapid movement down an electrochemical gradient driven by concentration and charge differences. Carrier proteins bind their specific molecule, change shape, and release it on the other side. This is usually slower.
And this movement can be passive or active.
Right. Passive transport uses the existing gradient, no energy required. Active transport moves things against their gradient, which requires energy, usually from ATP hydrolysis, primary active transport, or by coupling the movement to another ion flowing down its gradient, secondary active transport.
Like the sodium gradient used to pull glucose into intestinal cells.
Exactly. That relies on the NAP plus OK plus ATPase pump, a primary active transporter using ATP to pump sodium out and potassium in, maintaining that crucial sodium gradient. Another example is the MDR protein in cancer cells, an ATPase that pumps chemo drugs out, causing resistance.
And water movement is always passive, following the solutes.
Always passive, driven by osmosis. Cells have lots of charged molecules inside, drawing in cations and water. They constantly need to pump ions out, mainly sodium, via the NAB plus CoG-Mol plus pump to avoid swelling and bursting. If ATP production fails, the pump stops, ions and water rush in, and the cell is CS. Similar pumps maintain crucial gradients for calcium, hydrogen ions, pH, and others.
Okay, moving inside the cell now. The endoplasmic reticulum, ER. Rough and smooth. What are their roles?
The ER is this huge network of membranes throughout the cytoplasm. It's the main site for synthesizing transmembrane proteins, lipids for membranes, and proteins destined for secretion or other organelles like lysosomes. The rough ER, RER, is studded with ribosomes.
For protein synthesis, right?
Yes, specifically for proteins that are entering the secretory pathway or getting inserted into membranes. Ribosomes translating these proteins dock onto the RER, and the growing polypeptide chain is fed into the ER lumen, or embedded in the ER membrane. Proteins synthesized on free ribosomes stay in the cytosol.
And inside the RER, proteins get modified.
They fold into their correct shape, often helped by chaperone proteins. They assemble into multi-subunit complexes, disulfide bonds form, and they get their initial sugar tags and linked glycosylation. The ER has strict quality control. Misfolded proteins are held back and usually degraded.
Like the CFTR protein in cystic fibrosis.
Exactly. The common mutation causes misfolding, retention in the ER, and degradation, so it never reaches the plasma membrane. If too many misfolded proteins build up, it triggers ER stress and the unfolded protein response, UPR.
The cell tries to cope by making fewer proteins and more chaperones.
Right. But if the stress is too much, the UPR can trigger apoptosis, cell death.
Okay, that's the rough ER. What about the smooth ER, SER, no ribosomes?
Correct. The SER is more sparse in most cells, but prominent where specialized functions occur. It's big in cells making steroid hormones, gonads, adrenals. It's crucial for detoxifying lipid-soluble drugs, and the liver exposure can cause the SER to expand, called hyperplasia. It also stores and releases intracellular calcium, which is a key signaling molecule.
Like in muscle contraction.
Yes, the specialized SER in muscle is the sarcoplasmic reticulum, controlling calcium release for contraction. The SER is also involved in lipid synthesis.
After the ER, proteins and lipids move to the Golgi apparatus, the cell's post office.
That's a good analogy. The Golgi receives cargo from the ER, modifies it further, sorts it, and packages it for delivery. It's a stack of flattened membrane sacs called cisternae running from a cis face, receiving from ER, to a trans face, shipping out.
And proteins get modified as they move through.
Yes, sequentially. N-linked sugars from the ER are trimmed and elaborated. O-linked sugars are added. These modifications are important for protein function and sorting. For instance, adding a mannose-6-phosphate tag targets proteins to the lysosome.
And then, from the trans Golgi, they're dispatched.
Exactly. Sorted into vesicles heading to lysosomes, the plasma membrane for secretion or insertion or other destinations. Cells that secrete a lot, like plasma cells making antibodies or goblet cells making mucus, have very prominent Golgi.
Okay, biosynthesis covered. What about waste disposal? Lysosomes and proteasomes.
Two key systems. Lysosomes are membrane-bound sacs filled with about 40 different types of digestive enzymes. Acid hydrolysis that work best at low pH, around 5.
How do things get to the lysosome for degradation?
3 main routes. First, endocytosis brings material from outside the cell into endosomes, which mature and fuse with lysosomes. Second, autophagy, self-eating. The cell wraps up old organelles or damaged proteins in a double membrane, autophagosome, which then fuses with lysosome. It's crucial for turnover, survival during starvation, and clearing intracellular debris or pathogens. Third, phagocytosis, mainly by immune cells like macrophages, engulfing large particles like bacteria into a phagosome, which then fuses with a lysosome.
So lysosomes handle bulk degradation. What about proteasomes?
Proteasomes are large protein complexes in the cytosol nucleus. They primarily degrade individual cytosolic proteins that are damaged, misfolded, or just need to be removed as part of normal regulation, like transcription factors or cell cycle proteins.
How are proteins targeted to the proteasome?
They get tagged with a small protein called ubiquitin. Usually a chain of ubiquitin molecules is attached. This polyubiquitin tag is recognized by the proteasome, which unfolds the protein and feeds it into its central chamber, chopping it into small peptides. These peptides are then recycled. This is crucial for controlling levels of key regulatory proteins. It's also involved in antigen presentation for the immune system.
And briefly, peroxisomes.
Smaller organelles with oxidative enzymes. They're important for breaking down very long-chain fatty acids, generating hydrogen peroxide, H2O2 in the process, which they then safely neutralize with catalase.
Okay, a very sophisticated breakdown and recycling system. Let's not forget the mitochondria. More than just powerhouses.
Much more. They generate most of the cell's ATP through oxidative phosphorylation, yes. But they have their own DNA, mtDNA, inherited mostly maternally, and machinery resembling bacteria, reflecting their evolutionary origin as engulfed prokaryotes.
And they're dynamic.
Very. Constantly fusing and dividing fission for quality control. They have a limited lifespan, maybe 110 days, and get removed by mitophagy, a type of autophagy. Their structure inner membrane folded into cristae containing the respiratory chain, and the central matrix with metabolic enzymes is perfectly suited for energy generation.
They burn fuel completely for lots of ATP.
Right. But rapidly dividing cells, like cancer cells, often switch to less efficient aerobic glycolysis, the Warburg effect. They get less ATP per glucose, but generate building blocks needed for proliferation. Mitochondria are also central hubs for metabolism beyond just energy.
And they play a key role in cell death.
Absolutely critical. In necrosis, severe injury damages mitochondria, leading to ATP depletion and cell death. In apoptosis, programmed cell death, mitochondria integrate survival and death signals. If the death signals win, mitochondria release proteins like cytochrome C into the cytoplasm, triggering the caspase cascade that executes the cell. Controlling apoptosis is vital, Cordovi. Too little contributes to cancer, too much to neurodegeneration.
An incredible range of functions. Okay, let's move to how cells communicate and respond to their environment cell signaling.
Absolutely essential for multicellular life. Development, tissue function, responding to threats, it all relies on cells talking to each other. Disruptions lead to things like cancer or shock.
And cells are constantly integrating signals.
Constantly. They receive diverse signals, danger signals, signals from direct contact with other cells or the matrix, and secreted molecules like growth factors, cytokines, and hormones. The combination and intensity often determine the specific response. Differentiate, divide, perform a function, or even just survive. Lack of survival signals can trigger apoptosis.
How do these signals travel between cells?
Several ways. Paracrine signaling is local signals diffuse short distances. Autocrine is when a cell signals itself, maybe to amplify a response. Synaptic signaling is specialized for neurons releasing neurotransmitters. Endocrine signaling uses hormones traveling through the bloodstream to distant targets.
But regardless of how the signal travels, it needs a receptor on the target cell.
Yes, specific receptor proteins bind the signaling molecule, the ligand, with high affinity. Receptors can be inside the cell or on the cell surface.
Intracellular receptors for signals that can cross the membrane.
Right, like steroid hormones or vitamin D binding to nuclear receptors, which are basically ligand-activated transcription factors.
Wow.
Or small gases like nitric oxide, NO, diffusing into smooth muscle cells, activating an enzyme directly to cause relaxation.
And cell surface receptors for signals that can't get in?
Exactly. These are transmembrane proteins. Ligand binding triggers a change. It might open an ion channel, activate an associated G protein, activate an enzyme, often a kinase, linked to the receptor, or trigger protein cleavage or stabilization to activate a transcription factor.
So the signal gets relayed across the membrane. That's signal transduction.
Precisely. Ligand binding changes the receptor's shape, often causing receptors to cluster together. This change on the inside part of the receptor kicks off a cascade of intracellular events.
And there are different types of these surface receptors.
Several major classes based on their signaling mechanism. Many involve phosphorylation, adding phosphate groups to proteins catalyzed by kinases. Receptor tyrosine kinases, RTKs, have intrinsic kinase activity in their cytoplasmic tail, activated by ligand binding. Think receptors for insulin, EGF, PDGF.
And some receptors recruit separate kinases.
Yes, non-receptor tyrosine kinases. Receptors for many cytokines or immune signals work this way. Ligand binding causes them to associate with intracellular kinases like SHG, which then phosphorylate targets. These kinases often have domains like SH2 binds phosphotyrosine and SH3 binds proline-rich regions that help assemble signaling complexes.
What about G protein-coupled receptors, GPCRs?
Huge family, over 800 types. They have 7 transmembrane segments. Ligand binding causes them to interact with an intracellular G protein, triggering it to exchange GDP for GDP, which activates it. The activated G protein then modulates downstream effectors, often leading to changes in second messengers like cyclic AMP or calcium ions released via IP3.
And the nuclear receptors we mentioned earlier.
They're inside, binding lipid-soluble ligands. The complex then binds DNA and regulates gene transcription directly.
And other types based on protein interactions, like Notch and WNT.
Right. Notch signaling involves ligand binding on another cell, triggering cleavage of the Notch receptor. The intracellular piece travels to the nucleus to regulate genes crucial in development. WNT signaling involves WNT ligands binding fizzled receptors, a type of GPCR. This stabilizes a protein called leucinin, allowing it to enter the nucleus and co-activate transcription, also vital for development and tissue maintenance.
These pathways sound incredibly complex, not just linear chains.
Far from it, it's a network. Activating one receptor can trigger multiple downstream pathways. Kinases phosphorylate many targets. Phosphorylation can activate inactivate enzymes, change protein location, affect stability, trigger feedback loops. It's intricate.
And adapter proteins help organize this.
Yes, adapter proteins act like molecular matchmakers. They have domains like SH2, SH3 that bind specific motifs on other proteins, bringing signaling components together into functional complexes or nodes in the network. Signal transduction is really about information flowing through this network. Systems biology tries to map and understand this complexity.
And ultimately, many pathways lead to changes in gene transcription via transcription factors.
Very often, yes. That's how signals cause lasting changes. Transcription factors bind specific DNA sequences in promoters near the gene start, or distant enhancers. They recruit co-activators or co-repressors, chromatin modifiers, and the RNA polymerase machinery to turn genes on or off. Their activity is tightly regulated by things like phosphorylation, localization, and stability.
Okay, let's tie this back to a key group of signaling molecules, growth factors.
Growth factors stimulate pathways promoting cell survival, growth, and division. They bind specific receptors and influence genes needed to enter the cell cycle, avoid apoptosis, and synthesize the materials for growth.
But they do more than just growth.
Oh, yes. They also regulate migration, differentiation, specialized functions. They're key in tissue repair and regeneration. But if their signaling goes awry, too much factor, or constitutively active receptors or downstream pathways, you get uncontrolled proliferation. Many components of these pathways are proto-oncogenes. Mutations turn them into oncogenes driving cancer.
Let's run through some key examples. EGF, And TGFO.
Part of the EGF family, bind-related receptors, EGFRs, which are RTKs. Mitogenic for many epithelial cells, fibroblasts. EGFR itself, B1 and HRR2, URB2, are often mutated or amplified in cancers like lung, breast, brain. Drugs targeting them can be very effective.
HGF hepatocyte growth factor.
Also called scatter factor. Mitogenic for liver cells and most epithelia. Important in development, promotes migration. Its receptor is MET, another RTK often mutated in kidney and thyroid cancers. MET inhibitors are being tested.
PDGF, platelet-derived growth factor.
Comes in different forms, AA, AB, BB, released by platelets at injury sites, but also made by macrophages, endothelium, binds PDGFRs, alpha and beta, both RTKs, stimulates proliferation of fibroblasts, smooth muscle, endothelium, attracts these cells and inflammatory cells to wounds.
And VEGF, vascular endothelial growth factor, crucial for blood vessels.
Absolutely. VEGF-A is the main driver of angiogenesis, new blood vessel formation and development, wound healing, and tumors. Other VEGFs handle lymphatics or specific vessel types. VEGF promotes endothelial migration, proliferation, tube formation, and increases permeability. Hypoxia is a major trigger. It binds VEGFRs, mostly VEGFR2 on endothelium, which are RTKs. Anti-VEGF therapies are vital in treating some cancers and eye diseases like wet macular degeneration.
FGF, fibroblast growth factor.
Large family over 20. FGF1 acidic and FGF2 basic are well known.
Yeah.
Involved in wound healing, development, angiogenesis, they bind ECM components like heparin sulfate, creating a reservoir. Signal through FGFRs 1 to 4, which are RTKs.
And finally, TGFS, transforming growth factor beta. Seems complex.
Very complex, often called pleiotropic. 3 main isoforms, 1, 2, 3. Signal through type IN2 receptors, which are serine kinases, activating SMAD transcription factors. In wound healing, it strongly promotes scar formation, stimulates collagen production, inhibits its breakdown, crucial for fibrosis and chronic inflammation. But it's also anti-inflammatory, suppressing lymphocyte activity. Its effects really depend on context.
That's a great overview. Now, let's talk about the environment around the cells, the extracellular matrix, or ECM, more than just scaffolding.
Way more. It's this intricate network of proteins and polysaccharides that fills the spaces between cells. Yes, it provides mechanical support, anchorage for cells, tracks for migration, helps define cell polarity, but it also actively regulates cell behavior.
How so?
It binds and presents growth factors to cells. It signals directly through cell surface receptors like integrins. It acts as a reservoir for latent factors activated during injury. It's essential for tissue renewal. The basement membrane provides a template for regeneration, and it defines tissue microenvironments.
And it's dynamic, constantly remodeled.
Constantly. Synthesis and degradation are balanced during development, wound healing, but also dysregulated in fibrosis or cancer invasion, where cells need to break through it.
What are the main forms? Interstitial matrix and basement membrane.
Correct. Interstitial matrix is the filler in connective tissue between stromal cells or supporting epithelia. Made by fibroblasts, it's a 3D gel collagens, fibronectin, elastin, proteoglycans. Basement membrane is a specialized thin sheet underlying epithelia, endothelium, muscle, made by both epithelial and mesenchymal cells. Key components are type IV collagen and laminin. It's a selective barrier and organizing surface.
What are the main molecular building blocks of the ECM?
3 main families: fibrostructural proteins, collagens for strength, elastin for recoil, water-hydrated gels, proteoglycans and hyaluronin for resistant compression and lubrication, and adhesive glycoproteins, fibronectin, laminin, connecting components and linking cells via adhesion receptors like integrins.
Let's break those down. Collagens and elastin.
Collagens are the most abundant proteins, forming triple helices. Fibrillar dikes, I23V, form strong fibers in bone, tendon, skin, cartilage. Their strength comes from cross-linking, which requires vitamin C, hence scurvy if deficient. Genetic defects cause brittle bone disease or Ehlers-Danlos. Non-fibrillar collagens, like type 4, form the meshwork of basement membranes. Elastin provides elasticity, allowing tissues like blood vessels, lungs, skin to stretch and snap back. It forms fibers with a core of elastin surrounded by fibrillin glycoproteins. Fibrillin defects cause Marfan syndrome, weakening the aorta.
Okay. Proteoglycans and hyaluronin in the gels.
Proteoglycans have a core protein with long sugar chains called glycosaminoglycans. Gs attached like heparin sulfate, chondroitin sulfate.
Yep.
These GAGs are highly negatively charged, attracting water, forming hydrated gels that resist compression and lubricate joints. They also bind growth factors. Hyaluronin is a huge GAG itself, not attached to a protein core, also binds lots of water, contributing to viscosity and hydration.
And the adhesive glycoproteins and receptors, fibronectin, laminin, integrins.
These connect everything. Fibronectin links cells to collagen, fibrin, etc. via integrins, crucial in wound healing. Laminin is the major adhesive protein in basement membranes, connecting cells to type IV collagen. Integrins are transmembrane receptors on cells that bind ECM components like fibronectin and laminin, often via an RGD sequence. They link the ECM outside to the cytoskeleton inside, mediating adhesion and signaling, regulating migration, proliferation, differentiation. Crucial stuff.
The ECM is clearly a major player. Okay, final big topic. Maintaining cell populations. Cell growth, renewal, differentiation. Let's start with the cell cycle.
Right. Fundamental for development, tissue maintenance, repair. It involves replicating DNA accurately, S phase, growing, G1 and G2 phases, and dividing the chromosomes and cytoplasm, M phase. Non-dividing cells are in G0.
It's tightly regulated.
Very tightly. Progression depends on completing the previous stage. It's driven by proteins called cyclins, whose levels rise and fall cyclically, and cyclin-dependent kinases, CDKs, enzymes that are activated when bound by a cyclin. Different cyclin-CDK pairs control different transitions, like cyclin DCDK46 and cyclin ECDK2 for G1 to S, cyclin ECDK2 for S phase, cyclin BCDK1 for entry into mitosis.
And there are checkpoints, safety mechanisms.
Absolutely. Checkpoints monitor for problems like DNA damage or incomplete replication. The G1S checkpoint checks DNA integrity before commitment to replicate. G2M checks if replication finished correctly before division. If problems are found, the cycle halts for repair. If damage is too severe, it triggers apoptosis or senescence, permanent arrest.
How are checkpoints enforced?
By CDK inhibitors, CDKIs. Proteins like P21, P27 broadly inhibit CDKs. Others like P16, P15 specifically inhibit G1 CDKs, CDK46. They act as brakes. If CDKIs are faulty, damaged cells might divide, risking mutations and cancer.
And cell division isn't just about DNA. The cell has to grow too.
Right. Growth factor signals that push the cell cycle also activate metabolism to build new membranes, proteins, organelles. That switch to aerobic glycolysis, the Warburg effect, provides the building blocks needed for growth.
This regulation is complex. Now, what about stem cells? They're key for renewal.
Absolutely. Stem cells have two key properties: self-renewal, making more stem cells, and differentiation, producing specialized cells. Embryonic stem cells, for the blastocyst inner cell mass, are pluripotent, can become any body cell type. Adult stem cells reside in tissues and are usually multipotent or unipotent, replacing cells within that tissue lineage.
How do they maintain tissues?
Through a balance of self-renewal and differentiation. In high-turnover tissues like skin or gut, stem cells constantly divide. One daughter might differentiate, the other stays a stem cell, asymmetric division. Or both might remain stem cells, symmetric division, to expand the pool, especially during development or stress. This maintains homeostasis, balancing cell production with cell loss.
Where do adult stem cells live?
In specific microenvironments called stem cell niches. These niches provide signals that regulate stem cell quiescence, proliferation, and differentiation. We find them in bone marrow, hematopoietic and mesenchymal stem cells, intestinal crypts, hair follicles, cornea, brain subventricular zone, and elsewhere.
Hematopoietic stem cells, HSCs, make blood cells.
Yes, continuously replenishing all blood lineages. They're used clinically in bone marrow transplants.
And mesenchymal stem cells.
MSCs.
Also from bone marrow and fat. They can become bone, cartilage, fat, muscle cells. They're interesting for tissue engineering and because they seem somewhat immune privileged.
Which brings us to regenerative medicine, using stem cells to repair damage.
That's the goal. Theoretically, we could use stem cells or their progeny to repopulate damaged tissues like heart muscle or neurons, or even build replacement organs. Big challenges remain, though, getting cells to integrate properly, avoiding immune rejection, since most stem cells express HLA molecules.
Except maybe MSCs, or using induced pluripotent stem cells, iPSCs.
Right. MSCs might be less immunogenic, and iPSCs reprogramming a patient's own adult cells back into a pluripotent state offer a route to making patient-specific cells for transplantation without rejection. That's a major focus now.
This has been an incredibly thorough journey through the cell. We've gone from the genome's complexity and epigenetic control through gene editing, essential housekeeping, signaling networks, the ECM, and finally stem cells and tissue maintenance.
It really covers the core concepts, and as we've highlighted, understanding these molecular and cellular details is absolutely fundamental to understanding how diseases arise and how we might effectively treat them. Pathology today is deeply rooted in this molecular understanding.
It truly is, and as we wrap up, you can't help but think about the implications. The ethical debates around gene editing are only just beginning. And the potential to, say, manipulate stem cell niches to boost regeneration for currently incurable conditions, it's staggering.
It is. The pace of discovery is relentless. With greater understanding comes both immense opportunity and significant responsibility.
Absolutely. For anyone wanting to dive even deeper into these areas, definitely check out the references and resources associated with this deep dive. There's always more to learn. Thank you for joining us today on the deep dive.